Hi everyone, this is Dr. Barker. It looks like my recording from Monday failed, so I'm re-recording this a little bit later. I know that it won't match your notes exactly, but hopefully this will still be useful. So in our previous class, we had talked about the immediate innate immune response, which really was the humoral part of the innate immune response, or the proteins that were a major part of the innate immune response, the antimicrobial peptides and complement. These are able to work immediately because they don't require any sort of activation. And oftentimes they can recognize the pathogen, eliminate that pathogen, and end the infection. And then, of course, there will be tissue repair. One thing that I wanted to point out about this material is a point that I forgot to make last time about the immune system in general, and one principle that we're going to see throughout the semester. So when we talked about the alternative pathway, we saw this molecule C3 that had a strained thioester that was able to undergo spontaneous cleavage and attach to different surfaces, pathogen surfaces as shown here, or lots of self surfaces. And we talked about how all of our cells have complement inhibitors that are able to remove that bound C3. What I hope that you can see here is that this is somewhat strange in terms of use of resources. You might imagine that it would be more efficient to have this protein only binding to foreign molecules. The fact that we spend so much time making this protein only to inhibit much of it as it is binding to self cells may seem a little bit inefficient to you. You are going to see things that look a little bit like this throughout the semester, where you see situations where there are immune molecules that, because of the important evolutionary threat of infectious disease, have evolved to respond broadly or to many different types of cells in a way that may seem a bit inefficient. We may be wasting some resources as we try to get rid of those, and you might think that that's less than ideal, but this is one place where I want you to see that the immune system has evolved in response to pathogen threat, and it certainly is not perfect. Later in the semester, you're going to come across some phenomena that may feel odd to you because they seem so inefficient. And I want you to realize that's one of those places where you're going to really have to think about kind of how evolution can solve these problems. But really, our topic for today is what happens after this immediate innate immune response. What if the pathogen is not eliminated? Fortunately for us, we have additional parts of the immune system here listed as the induced innate immune response. But as you'll see today, as well as in the next class, this is really about the cells of the innate immune system. They start responding a little bit later because they take a little bit of time to become activated. And these are really the dominant immune responses within, say, the first 4 days following infection. And so here we might have that pathogen invade, and we might have now cells that are going to make some type of response. Largely, this is a process that you have already heard of before. This is the process known as inflammation. And throughout much of our discussion today, we are going to talk through the steps that you see on this figure. So we're going to go through this in some detail and break down the process that you see. There are a few things that you might think of as things that happen during the process of inflammation. They actually were first described by the Romans, so they have names in both Latin as well as the names that we know them as, and they're things that you can easily identify because you've probably experienced inflammation before. These are known as the 4 cardinal signs of inflammation, and they are redness or rubor, heat or calor, pain or dolor, and swelling or tumor. And so today you will see how those cardinal signs of inflammation are induced throughout this process. Oftentimes when people hear the word inflammation, if you think about the last time you heard the term inflammation on TV or something like that, it is used as a negative. People think about inflammation as being a huge cause of all sorts of problems, and you often hear about products that are trying to help you get rid of this terrible devil of inflammation. What I want you to realize first and foremost is that inflammation is an important physiologic process that has a key role. Inflammation is not always bad. If you have infection, tissue injury, or potentially tissue stress or malfunction, inflammation is the process that happens to allow you to have host defense, tissue repair, or adaptation and restoration of homeostasis. All of these are good things. There are ways that inflammation can go wrong, but at its sort of most basic level, inflammation is a very useful, beneficial, and adaptive process. The first cells that we are going to see acting in this inflammation process are monocytes and macrophages. You can see monocytes and macrophages shown here. The monocyte that you saw in last week's lab, as well as a macrophage that may have differentiated from a monocyte. And of course, that macrophage will live in tissue. It may be a microglia. If we're in the brain, it may be a Kupffer cell. If we're in the liver, it may be a Langerhans cell if we're in the skin. And so the first response that we are going to see is the response of one of these tissue macrophages. If you look at this process of inflammation, you can see tissue damage being induced by perhaps a piece of glass or something like that. And that allowing in microbes, in this case, bacteria. Much of this process, as I'm going to talk about it today, is classically thought of as a response to bacterial infection. I will point out that that is oversimplified. You can see some of these processes having a role against other pathogens too, but we are going to oversimplify and talk about this as a response to bacteria right now. We can see that tissue damage happening and bacteria coming into the system. And as the box in the upper left corner says, the tissue damage and bacteria will cause resident sentinel cells to release stuff. Those resident sentinel cells, in fact, are those resident macrophages that are in the tissue. In this case, it might be skin, so it might be those Langerhans cells. And they are going to do an action, this release stuff that is said in the rest of that box. In order to perform this action, those cells needed to know that the bacteria are present. And that will, of course, allow them to make some type of response. And so the first thing that we need to think about is how those macrophages in that site know that bacteria are present. This is an example of a phenomenon known as self-non-self recognition. So our cell, like the macrophage shown on the left, has a number of different receptors that can bind to biomolecules on the microbe that are non-self. They are part of the microbe, in this case a bacterial cell, and are not found on human cells. And so the macrophage has these receptors that can perform non-self recognition. There are many of these different types of receptors, as you can see on the right, that are going to recognize some type of component coming from the microbe. One major, or the main group of those receptors that we are going to talk about are a group of receptors known as PRRs. PRRs stand for pattern recognition receptors. And so our macrophages will have PRRs that allow them to bind components from the microbe. The examples shown on this slide all show PRRs that are on the surface of the macrophage. There are many important such proteins on the surface of the macrophage, although there are also PRRs in compartments inside of the cell, in the cytoplasm, and even in the nucleus, because all of those are places where microbes may be present and where we may need to perform surveillance. Each cell will likely have multiple different PRRs, as you can see in the macrophage on the right-hand side. These are sort of normal receptors that will make more sense when we talk about some other types of receptors later in the semester that are encoded by sort of your typical types of genes. And these PRRs respond to parts of microbes. Sometimes people talk about these receptors of the innate immune system as being a bit less specific than receptors in the adaptive immune system. And that is in some ways true, but the idea is what you can see in the upper left here, that we will have one receptor that binds to a molecule that is common to many different microbes. So one receptor may bind to a large class of microbes instead of being specific to individual microbes. That may involve a response to something like LPS, part of the gram-negative cell wall, or peptidoglycan found in the cell wall of many bacteria. The PRR that binds to a component like that will be able to recognize the broad group of organisms that has that particular molecule. This is really great because we can do broad surveillance for non-self components. It's also really important because most of these molecules are important structural parts of the microbe. They are broadly conserved and they are not easily alterable by mutation. So in some cases, you could imagine a microbe undergoing some mutation to sort of switch up its target so that it no longer bound to some receptor of the immune system. In the case of these types of targets, these are structures that the microbe really is going to have quite a bit of difficulty in changing. The microbe is not going to reinvent the structure of LPS or peptidoglycan, for example. And so this receptor will be able to kind of recognize the microbe without the microbe being able to avoid it by mutation. In my lab, we work on some receptors that bind to DNA, and the microbes are not going to reinvent the structure of DNA, for example. So as I mentioned, the molecules from the microbes that are bound here are bound by receptors called pattern recognition receptors. There are specific names for the patterns from the microbes as well. The microbial patterns, which are bound by pattern recognition receptors, or PRRs, are often referred to as PAMPs. PAMPs stands for pathogen-associated molecular pattern. And in this table, you can see a number of examples of PAMPs here. So you can again see peptidoglycan as well as triacyl lipopeptides. There's zymosan, there's LPS, there's flagellin, a number of different potential PAMPs. Many people in the field have started to change the term PAMP and use another term instead. Many people often call these molecules MAMPs, which stands for microbe-associated molecular pattern, because it has been realized that many of these molecules are found on microbes in general, not just pathogenic microbes. What you can see is the downstream result of what happens when a PRR binds a PAMP or a MAMP. There are a couple of different things that happen, one of which is shown here. So you can see that our macrophage has a number of different PRRs. Should one of those bind to a microbe, that will induce the phagocytosis of that microbe. So now that microbe will be internalized into a compartment inside the cell, and that microbe will be degraded. So one of the results of a microbe binding to a PRR is phagocytosis. You can see that same process of phagocytosis on the left-hand part of this slide, where we have a microbe binding to some sort of PRR, that microbe becoming engulfed and phagocytosed and then degraded inside of the cell. However, this is not the only thing that happens as a result of the microbe binding to the PRR. You can also see a signal transduction cascade, which is indicated here with a black arrow, that leads to changes in transcription in the cell. After we have a change in transcription, we will have new proteins produced by translation, and those proteins can then be secreted by the cell to have some type of physiologic effect. Most commonly, the proteins that we are going to talk about, at least today, are a group of proteins that are known as inflammatory cytokines. We are frequently going to see cytokines throughout this semester. Cytokines are proteins that are made by immune cells to allow for cell-to-cell communication, either between immune cells or between immune cells and other cell types. There are lots of different types of cytokines in the immune system. One subgroup of those is involved in inflammation. The ones that are involved in inflammation are called inflammatory cytokines, though you could imagine other types of cytokines existing as well. And so what we can see is that our receptor can lead to signal transduction that will change transcription and lead to the production of inflammatory cytokines. Much of the detail of that will be covered in the next lecture. In broad strokes, these are the final results of that signaling cascade. One thing that will happen is that we will see signaling from our PRR, and that will lead to enhanced ability for our phagocytic cell to kill the microbe. So the cell will become better at phagocytosis, better able to kill the microbe. We will also see inflammation, the rest of the process that we've been talking about, occur as a result of our inflammatory cytokines. This will also help the adaptive immune response, as we will talk about later in the semester. And finally, there will be some other types of tissue remodeling happening. So at this point, we've seen tissue damage, and we've seen those sentinel cells, or macrophages, release our inflammatory cytokines. And now our inflammatory cytokines are actually going to have some type of effect. We can see 3 really famous inflammatory cytokines that are made during this process. One is called IL-1 beta, one is called TNF-alpha, and one is called IL-6. And all 3 of those are going to have some actions that you see on the next slide. We also see some molecules made known as chemokines. One important chemokine that's made here is called IL-8. It's also known as CXCL8. Chemokine is a word that was made up by smushing together two words for a part of what this molecule does. Chemokines are chemotactic cytokines. They are cytokines that induce cell movement. And so chemokines are specifically important for attracting cells and allowing them to move to different locations. So not only do we see inflammatory cytokines being made in this process, we also see the production of some chemokines. Those inflammatory cytokines and chemokines have important effects. First, if you look at the blood vessel on the figure on the left, you can see that it's somewhat smaller in diameter. Once the inflammatory cytokines are made, that blood vessel increases its diameter. That is known as vasodilation. This will allow more blood to go to this site, including both more immune cells and more of our antimicrobial peptides, complement, and other potentially important proteins. You can also see that the orange cells that make up the wall of the vessel go from being tightly attached to one another to having gaps between them. That is known as increased vascular permeability, and that will allow fluid, cells, and protein to go into that site of infection. Finally, those chemokines will also attract other cells to come into this site of infection, as you see happening in this process. All three of these events are really important in resulting in the hallmarks of immunity that we mentioned. The increase in fluids and proteins and blood flow to this area can lead to both increase in redness and heat. You'll also see an increase in swelling because of that fluid and protein going to the area. One of the hardest parts of this to really explain is an understanding of what is going on with the increase in pain that is happening, the fourth of our hallmarks of inflammation. There have been a number of recent studies that have helped us understand how pain can be related to these inflammatory cytokines as well. This is one recent review that mentions ways that cytokines can be really important in leading to pain that can be seen as a result of an inflammatory response. I always sort of smile when I see this because I was often confused about how pain as well as how itch might play a role in inflammation and how that really tied into our inflammatory cytokines. It turns out one of the people who does some really lovely research on this is someone named Isaac Chu, who was one of my classmates in graduate school. So happily, Isaac started to figure out the answers for my confusion. You can also imagine this pain having an important evolutionary role, whereas when you have some type of wound, because that wound is in pain, it helps to alert you and helps you to protect that from further damage instead of, say, allowing further infection or further injury to that site. Because you have some pain, you will feel you'll be able to protect that site a bit better. One other thing that I want to point out here has to do with something we talked about in the previous class. So previously we saw two molecules, C3a and C5a, that were part of the complement cascade. And I told you that those two proteins were known as anaphylatoxins, which can lead to changes in vascular permeability and lead to some inflammatory responses. C3a and C5a, in fact, act a lot like inflammatory cytokines and chemokines and have many of the same effects that you saw on the previous slide. So this is one place where the immune system does not sort of behave nicely in terms of the different types of divisions that we're trying to make, because in fact these complement proteins, C3a and C5a, are acting very similarly to the cytokines that are made by the cells of the innate immune system. Those cytokines that are allowing chemotaxis, so the chemokines, are also very important for the recruitment of other immune cells to this site of infection where they are able to get rid of the microbes. There is a three-step process that is involved in the recruitment of these cells. This three-step process is similar for the recruitment of many types of immune cells to many different sites throughout the body, including movement of T cells to different sites, movement of inflammatory cells that we see here, among many others. We are going to go through these three steps when we talk about T cells later on in the semester, but those same steps are important here. In the case of inflammation, largely the cells that are being recruited to this site are neutrophils. And so we're seeing neutrophils get recruited to the site in order to get rid of the microbes. You can kind of imagine this like calling in the cavalry or calling in the SWAT team. And so the chemokines are going to be incredibly important for recruiting and attracting all of those other cell types. If you like the comics that I have in a few places in today's lecture, they are all from a site called Pageromics, which you can find online. All of the details that I have told you about so far when talking about inflammation are parts that are known as local inflammation. So we will see a situation where we have some tissue, perhaps your hand, that has an injury and that starts to have this type of inflammatory response. You will see that the macrophages in that tissue will start to secrete cytokines. We will see changes in the vasculature in that local area, like the hand, where we will see inflammation. So you can see the hand is red. Eventually, we're going to see those bacteria become phagocytosed. We're going to clear up that response and we'll live happily ever after. There are some other types of effects of some of our inflammatory cytokines as well. So some of them will act on other parts of the body after moving through the bloodstream. One example is that IL-1, IL-6, and TNF can act on the liver. And this will result in the liver making new proteins. One protein that will be made by the liver will be mannose-binding lectin. That will be an important part of the complement pathway that we learned about last time. And so again, you can see some interconnections between some of the different pathways that we're talking about. So these cytokines will actually improve the complement response by allowing more complement proteins to be made. We'll see things like fibrinogen being made to allow for coagulation and tissue repair. We'll also see some other proteins being made called C-reactive protein. All of this together is known as the acute phase response. And one of the reasons why I mention this is because sometimes when you go to the doctor to have blood work done, your C-reactive protein levels will be measured, which will be an indicator of what type of inflammation or whether you have inflammation occurring. Further, those inflammatory cytokines are able to act on the bone marrow. There are large numbers of neutrophils that are stored in the bone marrow, and when those inflammatory cytokines act on the bone marrow, those neutrophils are released so that there are larger numbers of neutrophils in the blood that can start to act against the microbe. You also may, in your blood works, have numbers of neutrophils measured, and that may be a way to, again, determine whether or not inflammation is happening. One other big thing that will happen as a result of inflammatory cytokine activity will be some type of interaction with those cytokines with the brain, particularly the hypothalamus, as well as with fat and muscle. And that will involve changes in metabolic processes and also a change in a homeostatic set point to increase body temperature. And that increased body temperature can function in order to decrease viral and bacterial replication. You have, of course, heard about this increased body temperature that happens as fever. So fever is not a response of is not a process that microbes are doing to you. Fever is actually something that your immune system is doing in order to stop microbial replication. Of course, if fever goes on for a long time, then you will see some bad effects as your cells are not going to do particularly well at high temperature either. But that fever is, in fact, an important response of your immune system. And so overall, those inflammatory cytokines will lead to that acute phase response, mobilization of cells from the bone marrow, as well as actions on the brain that may result in fever. You also can see things like sleepiness, lethargy, muscle pain, lack of appetite, nausea, all of those things that could happen as a result of our inflammatory cytokines. So many of the effects that you see when you feel ill are actually not the result of the microbe, they're the result of the immune system leading to a state where microbes can be stopped. This often leads students to ask a question about whether or not they should block fever. This is a New York Times article. It doesn't particularly come to a great conclusion. As I mentioned, people sometimes want to know whether or not they should stop taking medicines to get rid of their fever and let the fever go because it's actually part of their immune response and helps them. And my answer is, depends on the situation. And that's kind of what the New York Times comes down to as well. Recent studies, like this one in early 2019, have even given us more information about how fever is helping the immune system. And so there's still quite a lot that we can learn about this entire process, even in 2019. Once our cytokines have been made, that will change blood flow and capillary permeability. We can see an influx of fluid and cells into that site. And finally, we need to have these cells that have migrated to the site of inflammation, our neutrophils, actually destroy the bacteria. And so we finally need to talk about how those neutrophils are going to destroy the bacteria. Again, neutrophils are largely going to destroy incoming microbes by phagocytosis. So microbes will first bind to PRRs on the membrane. There will be quite a lot of cytoskeletal rearrangement so that the cell is able to reach around and make a grab of those microbes. This process, as I said, is called phagocytosis. And once the microbe is completely inside the cell, we call the vesicle or organelle where it is located the phagosome, because that organelle was made by phagocytosis. The phagosome will fuse with other components of the cell, other compartments inside the cell, which will deliver some other proteases and other types of molecules to help degrade our microbe. And we will also see increasing acidity throughout these compartments. So the microbe will be killed by different types of enzymes as well as low pH, and eventually we will degrade that completely. Of course, it's fun to see this in cartoon form, but it's much more interesting to see this process in action. This is a video that you can find on YouTube. It's actually on YouTube multiple times, put to multiple types of music. It's quite famous in the immune system. And what you're able to see is a neutrophil, which is this large cell in the middle, that is going to be getting rid of and chasing this bacteria, which is some staph. You can also see a few of these other smaller cells in the preparation. They're red blood cells, and they were really just contaminants in the preparation, but they do kind of look fun here as little types of obstacles. And so if you watch, we can watch our neutrophil actually dealing with this microbe. So you can see our neutrophil is able to track that microbe. It will follow it. And eventually our neutrophil will perform phagocytosis and engulf that microbe. One thing that I'd like you to notice happens earlier in the video. So if we watch our neutrophil moving here, you can see that our neutrophil is going to go after this particular staph organism, but you will see that it's also going to go very close to this other organism. It's not able to make changes to its entire cytoskeleton and switch which microbe it goes after, because there is quite a lot of cell biology going on in this process, and so you see that here. Once the process of phagocytosis has happened, you can see that our microbe will be in the phagosome. There will be many different types of granules inside of our neutrophil. Remember that our neutrophil is in fact a granulocyte, which means it's full of many granules. And those granules will fuse with the phagosome, providing many different types of enzymes that can kill the microbe. There also will be fusion with lysosome as the region gets more acidic. And so eventually we will see that there is full degradation of our microbe down to component parts like carbohydrates, lipids, sometimes individual amino acids, things like that. One of the ways that that microbe is degraded in the phagosome is by a process known as respiratory burst. There are enzymes within the phagosome that allow for the production of molecules like the superoxide anion, the hydroxyl radical, hydrogen peroxide, and hypochlorous acid, aka bleach, that are able to kill microbes and that are very antimicrobial. These in general are known as reactive oxygen species. Similarly, there are enzymes that allow for the production of nitric oxide, nitrogen dioxide, and peroxynitrite, which are reactive nitrogen species that also have antimicrobial effects. This is happening inside of the phagosome and the lysosome because we want to keep these toxic molecules in that compartment and not allow them to destroy the rest of the cells. So this process of respiratory burst is an important way that those microbes can be destroyed after phagocytosis. In addition, many of the compartments inside of our phagocytic cell will have many of the antimicrobial peptides that we discussed in the last class. So we will see things like defensins, like lysozyme, all being produced inside of the phagosome of our cell. And so again, we see some relationships between the different types of responses that we're talking about. One thing that is really unique about our neutrophil is that our neutrophil is quite short-lived. We talked a little bit about both macrophages and neutrophils in this class, and what you will notice is that they have some similarities and differences that you've seen. Both of them are able to do phagocytosis and destroy microbes. However, macrophages had the additional function of having the PRRs and leading to inflammatory signaling. Neutrophils largely just do phagocytosis, and then they will end up dying, whereas the macrophage is living longer to produce those inflammatory cytokines. As you'll see on the next slide, there may be one other new function of neutrophils, but the classic idea of a neutrophil is that it is going to die quite quickly after phagocytosis. You actually know a fair amount about the death of neutrophils because groups of dead neutrophils make a substance known as pus, which you have probably seen when you've had some type of cut before. And so what we frequently see is that we'll have our microbe phagocytosed by the neutrophil and degraded. Once that happens, our neutrophil will die, and some type of macrophage will perform phagocytosis and internalize that dead neutrophil, and that dead neutrophil will now provide nutrients for the macrophage. And so, as I mentioned, one characteristic of neutrophils is that they are very short-lived cells that we do not see living for a particularly long time. More recently, we have realized that neutrophils may have one additional function. This is the production of something known as neutrophil extracellular traps or neutrophil nets. You can see that the use of neutrophil here gets a bit redundant, as it's in both the abbreviation and the word you say before the abbreviation. There has been some controversy about how this is related to neutrophil cell death, whether the neutrophil is required to die in this process. And so that's one reason why I mention it when I'm talking about the short-lived nature of neutrophils. The process of making nets is often referred to as NETosis. And so in this process, our neutrophil will in fact release large amounts of cytoplasmic components, most of which include DNA. And this will make a sort of net that is able to trap the microbe, as you see at the top of this slide. And so the neutrophil is going to expel a lot of its nuclear components, which will lead to the production of this sort of trap that can trap many different microbes and stop them from being able to further replicate and move on. And in the bottom, you can also see a net coming out of a neutrophil. So this process of neutrophils expelling their DNA through NETosis is very important for making a sort of sticky matrix that can trap pathogens and keep them from further replicating and potentially further causing harm. And it's a really interesting sort of new function for neutrophils that has been described. All of the things that we've talked about thus far are parts of the beneficial side of inflammation, this great adaptive physiologic process that helps us for host defense, tissue repair, and maintenance of homeostasis. However, inflammation can sometimes go badly as well. If there is too much inflammation, if inflammation is happening in the wrong place, we can start to see things like autoimmunity, inflammatory disease, fibrosis, tumor growth, shift in metabolic set points, and a number of different pathological consequences. So inflammation that is controlled the right amount at the right time in the right place is good. Inflammation that is too much in place or time or amount will be a big problem. One example of this is this process known as septic shock. This happens when we see inflammation happening throughout the body instead of at one local site. So in the previous slide that looked like this, you saw inflammation happening in the hand. Perhaps we had a tissue injury in the hand where there were some microbes there and we were able to see an inflammatory response there. We also could imagine a situation where there was a systemic infection with bacteria, so that bacteria was in the blood instead of in one specific location. As a result, we are going to have macrophages throughout the body in the blood making a response and producing inflammatory cytokines. This is in contrast to the other example where we had macrophages in the hand making this response. Because we have macrophages making inflammatory cytokines throughout the body, the vasculature throughout the body will be changed. So we're going to see vasodilation and changes in vascular permeability throughout the body instead of in one site. There's going to be fluid and cells leaking out of the bloodstream throughout the body instead of at one site. As a result, we have systemic edema or systemic swelling happening everywhere because fluids are leaving the blood vessels. As a result, there won't be enough stuff left in the blood vessels and we'll have decreased blood volume. This may lead to lower levels of protein, lower levels of cells in the blood, all of which are bad. This can even lead to collapse of blood vessels because there's not enough blood volume inside the vessels to hold them open. Eventually, this may lead to problems in clotting throughout the body, things like wasting and multiple organ failure, and may lead to death. This is a common cause of death in hospitals and is one of the reasons why we find it so important to understand this inflammatory process. We can also realize that if we have excess amounts of these cytokines working on some of our different organs throughout the body for long periods of time in excess, we may see things like changes to the heart, where we'll change heart output and have cardiac issues. We may see changes to the vasculature in terms of increased permeability or thrombus, where we again are going to see cardiovascular problems. We could also imagine that if we have long-term changes to the metabolism of muscle, we may see things like insulin resistance. And so we are learning more and more that chronic inflammation, production of these cytokines for too long or at too high levels can lead to a number of different conditions. They may lead to tissue changes like cell death, like fibrosis, like changes in blood vessel growth in the wrong place, cell proliferation, eventually leading to heart disease, organ failure, cancer, Alzheimer's, autoimmune disease, autoinflammatory disease, inflammatory bowel disease, allergy, diabetes, impaired insulin signaling, obesity, among many others. We're still not completely sure what causes this chronic inflammation. Some of it may have to do with the genetics of some of the genes encoding proteins that are important in the immune system. Some may have to do with infections that don't go away or an inappropriate response to commensal microbes. There are also things that have been realized, like the fact that certain types of lipids lead to signaling through PRRs, and so things like obesity or excess lipid may lead to chronic inflammation, among many other sources. More and more, we are finding many disease states that are linked to chronic inflammation, which is why a really good understanding of this process is so key for many parts of medicine. We will continue to talk about that in the next lecture. I'm sorry that this is not a traditional lecture, but hopefully this was useful. Thank you.